Welcome to our lecture on aminoacyl-tRNA synthetases. In the microscopic world of your cells, translating the genetic code into physical proteins is a monumental task. Imagine running a highly specialized factory where a single mistake could ruin the entire product. Today, we are going to meet the brilliant, incredibly strict managers of this cellular factory. These enzymes are the true decoders of the genetic code, ensuring that every single building block is perfectly matched before the assembly line even starts. Let us dive in and discover how they achieve such absolute precision. Now that we know who we are talking about, let us understand their role through a simple analogy. Imagine you are running a highly specialized translation agency. In this agency, you have specific workers called tRNAs, and each worker is only allowed to carry one specific type of building block, which we call an amino acid. But how do you guarantee that the right worker always picks up the exact right building block? You hire a brilliant, incredibly strict manager whose only job is to check the worker's identification and hand them the correct block. In your cells, this strict manager is the enzyme known as aminoacyl-tRNA synthetase. Building on that analogy, let us look at the actual biological process. Formally, an aminoacyl-tRNA synthetase, often abbreviated as AARS, is a specific type of ligase enzyme found in all living cells. Its primary biological function is to catalyze the covalent attachment of a specific amino acid to its corresponding, or cognate, transfer RNA molecule. This crucial attachment process is scientifically referred to as charging the tRNA, or aminoacylation. Because there are 20 standard amino acids used to build proteins, a typical cell contains exactly 20 distinct types of these synthetase enzymes, one dedicated to each amino acid. Each specific synthetase recognizes the unique structural features of its target tRNA, ensuring absolute precision. Understanding how these enzymes charge tRNA brings us to a critical vulnerability in the cell. The ribosome, which is the cell's protein factory, is essentially blind to the amino acid itself. It only reads the anticodon on the tRNA. If an aminoacyl-tRNA synthetase makes a mistake and attaches the wrong amino acid to a tRNA, the ribosome will unknowingly incorporate that incorrect amino acid into the growing protein chain. This single error can cause the resulting protein to misfold, malfunction, or even become toxic, which is the root cause of many severe genetic and neurodegenerative diseases in humans. Therefore, these enzymes possess built-in proofreading mechanisms to double-check their work. To truly appreciate how accurate these enzymes are, despite the blind ribosome, we must look at the mathematics of enzyme kinetics. The synthetase must choose the correct amino acid out of a pool of very similar molecules. The error frequency, represented by the variable f, is determined by the ratio of the catalytic efficiency for the incorrect substrate to the catalytic efficiency for the correct substrate. In this equation, Kcat represents the turnover number, which tells us how fast the enzyme processes the substrate. The term Km is the Michaelis constant, representing the enzyme's affinity for the substrate. When the enzyme's affinity for the correct amino acid increases, the denominator becomes much larger, which drastically decreases the overall error weight, F. Now that we have our kinetic fidelity equation, let us apply it to a real biological scenario. Let us imagine we are studying the enzyme isoleucyl-tRNA synthetase, which must attach the amino acid isoleucine to its tRNA. However, the amino acid valine looks almost identical. Laboratory experiments show that the catalytic efficiency for the correct amino acid, isoleucine, is 10,000 units, while the efficiency for the incorrect valine is 50 units. We substitute these given experimental values into our error frequency equation. By dividing 50 by 10,000, we arrive at a final calculated error frequency of 0.005. This means the enzyme makes 5 mistakes for every 1,000 amino acids processed. This calculated error rate of 0.005 leads us to a fascinating evolutionary adaptation. Making 5 mistakes for every 1,000 amino acids processed might sound small, but because proteins are large and complex, this specific error rate is actually far too high for long-term cellular survival. It perfectly explains why these enzymes could not rely on initial binding alone. To survive, they evolved a secondary editing site. This editing site acts as a second sieve to catch and destroy these specific mistakes before the charged tRNA ever reaches the ribosome, ensuring the final error rate drops to near zero. Having seen the extreme lengths the cell goes to for accuracy, we must now consider the metabolic price. The process of charging a tRNA is not free. It requires a significant investment of cellular energy in the form of ATP. During the charging reaction, one molecule of ATP is broken down into AMP and pyrophosphate. This specific chemical breakdown is equivalent to consuming 2 high-energy phosphate bonds for every single charging event. The cell is completely willing to spend enormous amounts of its hard-earned energy to ensure that translation is highly accurate, highlighting a fundamental biological principle regarding cellular priorities. To quantify this metabolic burden, we can express the energy investment as a simple linear equation. The formula is E equals N times 2. In this equation, the variable E represents the total energy cost, measured in high-energy phosphate bonds. The variable N represents the total number of amino acids that make up the target protein we want to build. The constant 2 represents the 2 phosphate bonds that are permanently broken when a single ATP molecule is converted all the way down to AMP during a single charging event. If the length of the protein N increases, the total energy cost E will scale up in a perfectly linear fashion. Let us put this energy formula to work with a practical example. If a human cell needs to synthesize a specific protein that is exactly 450 amino acids long, how many high-energy phosphate bonds are consumed solely by the aminoacyl-tRNA synthetases? We begin our calculation by substituting our specific protein length into the equation, replacing the variable N with 450. We then multiply this value by the constant 2 to account for the dual phosphate bond energy cost per single amino acid. The final calculation reveals that exactly 900 high-energy phosphate bonds are consumed just to charge the tRNAs for this single protein. Now that we have used this formula to find the total energy cost, let us formally derive where it comes from, starting with the chemistry of a single reaction. This chunk of the proof establishes the baseline energy requirement. We know that a single charging event requires 1 molecule of ATP. During the reaction, ATP is hydrolyzed into AMP and a pyrophosphate molecule. Because the ATP loses 2 phosphate groups instead of just 1, this specific hydrolysis breaks 2 high-energy phosphoanhydride bonds. Therefore, we can mathematically state that the energy cost per single amino acid is exactly 2 bonds. This establishes the constant multiplier in our equation. Building on the baseline cost we just established, we can now scale this up to the entire protein. This final chunk of the proof connects the individual reaction to the macroscopic biological product. We know that each amino acid must be attached to a tRNA before it can be added to the growing protein chain. Therefore, the total number of charging reactions is exactly equal to the number of amino acids in the final protein structure, which we call N. By multiplying the total number of reactions, N, by the cost per reaction, which we proved is 2, we arrive at our final derived formula, E equals N times 2. To visualize everything we have discussed, let us look at this detailed schematic of the tRNA charging mechanism. On the far left, you can see the amino acid and the ATP molecule entering the active site of the aminoacyl-tRNA synthetase enzyme. Notice how the ATP is broken down, releasing the pyrophosphate molecule, shown in red, which represents the massive energy investment we calculated. This creates an intermediate state. Then, the uncharged tRNA molecule, shown in green with its distinct cross-like structure, enters the enzyme. The enzyme transfers the amino acid onto the acceptor stem of the tRNA. Finally, on the right, the fully charged tRNA is released, ready to travel to the ribosome. We have covered a lot of ground today, so let us summarize the most important ideas. First, aminoacyl-tRNA synthetases are the absolute gatekeepers of genetic translation, meticulously pairing each of the 20 amino acids with their correct tRNA molecules. Second, we saw mathematically how precise kinetic recognition, backed up by built-in proofreading editing sites, maintains an incredibly low error rate, protecting the cell from toxic proteins. Finally, we learned that this extreme accuracy comes at a heavy metabolic cost, consuming massive amounts of cellular energy. You now understand that without the strict oversight of these enzymes, the genetic code would be meaningless. Great job today.